Have you ever just stopped and wondered how the information crammed inside your DNA, this molecule with billions of letters, actually builds you? Well, today we're going to unlock that secret code. We're diving into the fundamental process that turns your genetic blueprint into a living, breathing being. Okay, let's get right into it. I mean, it sounds kind of impossible when you think about it, right? The entire instruction manual for a person or a tree or even a tiny little bacterium is written in an alphabet that only has 4 letters. So how in the world does this super simple code create the mind-boggling complexity of life? That's the puzzle we're going to solve together today. All right, to get started, we have to go to the source of it all. We're talking about the master blueprint for every living thing, the one and only DNA. Now, this massive DNA blueprint isn't just one long chaotic sentence, not at all. It's actually neatly organized into chapters, and we call those chapters genes. You can think of each gene as a single recipe in a giant cookbook. And what's it a recipe for? One specific thing, a protein. And it's these proteins, these little workhorses, that do pretty much everything, determining your eye color, your blood type, you name it. You know what? Let's make this even simpler with an analogy. Imagine your cell is a huge construction site. The DNA? That's the master blueprint. It's so precious, it's locked away for safekeeping in the main office, which is the cell's nucleus. Now, to get the plans to the workers, you need a temporary copy, right? That's RNA. And the final thing you build out on the factory floor, that's the protein. Keep this analogy in your head, because it's going to be our guide through this whole process. So we've got our blueprint locked up tight in the nucleus, but the factory floor, where all the action happens, is outside. And that, right there, presents our first major problem. Think about it. You wouldn't take the one and only master plan for a skyscraper and drag it out onto the muddy, messy construction site, would you? Of course not. It could get wrecked. Well, the cell thinks the exact same way about its DNA. It is the master copy, and it has to be protected at all costs inside that nucleus. So how do you get the instructions out to the workers? The solution is actually pretty genius. You make a copy. This process is called transcription. Basically, the cell makes a temporary, disposable copy of a single gene, and this copy is a molecule called RNA. It's literally like photocopying one page of the blueprint and sending that out to the factory floor, while the original stays perfectly safe back in the office. So how does this photocopying actually happen? Well, it's kind of like a little 3-step dance. First, the specific part of the DNA that's needed unwinds itself. Then the cell's machinery swoops in and makes a matching RNA copy, getting the code down letter for letter. And finally, this brand new RNA copy just peels away, zips right out of the nucleus, and makes a beeline for the factory floor. Okay, so our RNA photocopy has arrived, but its message is written in this weird 4-letter language. How does the cell's machinery have any clue how to read it? The genetic alphabet is incredibly simple. It only has 4 letters, or bases. In our RNA copy, those are A, C, G, and U. The entire instruction manual for all of life is written with just these 4 characters. So here's the puzzle that early life had to solve. You've got 20 different building blocks, amino acids, that you need to build proteins with. If you use a 1-letter code, well, you can only specify 4 of them. That's a non-starter. You're 16 short. All right, so what if we try a 2-letter code? That gets us 16 different combinations. We're getting closer for sure, but that's still not enough to cover all 20 of our building blocks. So nature came up with a better solution, reading the code in words made of 3 letters. 3. That's the magic number. And that 3-letter word completely solves the problem. A 3-base code gives us 64 possible combinations. That's way more than enough to give a unique code to each of the 20 amino acids. And you even have some codes left over for punctuation, like start and stop. And this 3-letter word, it has a name. It's called a codon. This is the fundamental unit, the basic word of the genetic code. When the cell's machinery reads that RNA strand, it reads it 1 codon at a time. GUA, then CCG, then AUU, and so on. And each one of those codons calls for one specific amino acid. Okay, we've got our instructions and we've cracked the code. Now it is time to head out to the factory floor and actually build our final product. This final stage is called translation, and it's pretty much what it sounds like. It's where the language of genes, which is written in nucleic acids, gets translated into the language of proteins, which is written in amino acids. This all goes down outside the nucleus, in the cytoplasm, at this amazing little molecular machine called a ribosome. And here is where the real magic happens. The RNA photocopy docks with a ribosome. That's our protein building factory. The ribosome then just chugs along the RNA strand, reading each three-letter codon. And for every single codon it reads, the exact right amino acid is brought in and linked onto a growing chain. This keeps going and going until it hits a stop codon. Then it releases this brand new chain, which folds up into a working 3D protein. And that new protein, it goes off to do its job. So, for example, if the gene was for the protein that makes melanin pigment, that protein will start cranking out melanin. If it makes a lot, an animal gets dark fur. If it makes just a little, the fur is light. A single gene, transcribed and then translated into a single protein, creates a physical trait you can actually see. This whole system is, it's just incredibly precise and elegant. But what happens when there's a typo in the blueprint? What happens when the code breaks?
Let's look at a real-world and very serious example: sickle cell anemia. The cause is a shockingly small error. In the DNA blueprint for hemoglobin, that's the protein that carries oxygen in your blood, a single letter T is accidentally switched to an A. Just one swap. That's it. This changes the RNA codon from GAA to GUA, a tiny one-letter typo.
But that one typo kicks off a devastating domino effect. The single letter change in the DNA leads to a different codon in the RNA. The ribosome reads this new codon, and instead of the right amino acid, it puts in the wrong one. Because of that one mistake, the entire hemoglobin protein folds up incorrectly. And that in turn warps the whole red blood cell into a stiff, fragile sickle shape. Just let that sink in for a second. A single incorrect letter in a genetic code that is 3 billion letters long, and the result is a painful, life-altering disease. This shows you, in the most powerful way possible, just how absolutely critical the accuracy of this entire DNA to protein process really is. And here's the final, and maybe the most amazing thought of all. The code we've just been talking about, the codons that stand for each amino acid, it's the same for pretty much every single living thing on this planet. The codons that build a protein in your cells are the exact same ones that build a protein in a bacterium, or a mushroom, or a whale. We are all reading from the very same genetic codebook. So what does this profound, deep biological unity tell us about our planets and our own shared history?